participate in NeuroHackademy.
Sorry about that.
No worries. It is always a delight to meet everybody at NeuroHackademy. I'm very much looking forward to when we can go back to doing this in person. But in the meantime, I'm just really enthusiastic about getting to know some of you and hearing about the ideas that you may have for how meta-analysis may be relevant to your own research. As I mentioned in the talk, meta-analysis is a really good introductory type project. It familiarizes you with the literature. It lets you really kind of dig in and get your hands dirty for some folks for the very first time. And it prepares you for that next level of your own study where you ask really novel, really unique questions. I am delighted that Taylor is joining us here. Taylor is the true master behind the NIMARE project. He has driven it single-handedly, and we are really, really excited to see how much progress that he has made. So with that, we're happy to invite any initial questions or comments about the lecture. And then as Ariel mentioned, Taylor does have prepared a really nice tutorial for how NIMARE works that you'll get to participate in. So any questions? I always love that empty little silence while we.
Maybe I'll warm us up with something here. So one thing, I think it came up in various ways in your presentation, but I thought I'd ask anyway more explicitly, which is something that we think about a lot in diffusion MRI when we combine different data sets, is what we call harmonization in diffusion, which is just the fact that the instruments are different, so they're measuring slightly different things. And when doing meta-analysis in functional MRI, I wonder how people think about things like, oh, this study was done with this resolution and this SNR, and this study over here was done with this resolution, this SNR.
So we do want to make sure we focus on the difference between mega-analysis and meta-analysis, right? And so when you think about harmonization, usually you're trying to think about mega-analysis, where you're combining the actual fMRI data, you're combining the DTI data that's acquired at different sites. Meta-analysis is somewhat less challenging and somewhat then more problematic, because we're not actually combining that raw data, we're combining the summary statistics. So in terms of the harmonization, it's really easy to take sets of coordinates that all refer to, and I'm going to air quote, the same standard template, and pool them to look for convergence. But once you really start thinking about what you're doing, different scanners, different analysis procedures, like the NARP study showed us. There are different ways of thresholding p-values, different whole kinds of different ways that people do everything. All of that gets loaded into the meta-analysis and becomes basically noise, right? And so some folks, that's an immediate turnoff and makes them go, this is not telling us really anything. I have made my peace with it by saying that, through the noise does emerge some signal. We have done enough meta-analyses that they are informative in telling us what are the most consistent effects that can be observed when you consider all of that variability. And that does tell us something about the literature. That does tell us something about all of the different studies that a group of domain experts have done in the field of working memory. And it certainly absolutely doesn't replace the precision and the deep knowledge that you can get from a single study. But particularly in the case of developing neuroanatomical hypotheses, it does set you up and say, look, 30 studies have all used roughly a similar paradigm. And even though they've used really different analysis methods and, are all at different sites and use different participant samples, the medial prefrontal cortex is really emerging as a significant region of interest. And so this meta-analysis is going to demonstrate that I really need to kind of start my inquiry in this particular region. So it's a tool. It's like any others. It's a flawed tool. And the deeper you dive into, the nuance of actually conducting your own meta-analysis, it's a pretty deep dive into those flaws. But it is so useful for doing a really important thing, which is allowing us to take a step back and really get a bird's eye perspective of what have we done as a community. Like what I like is that we're sort of standing on the shoulders of the giants who have come before us. And if we don't do meta-analysis, then we're acting like we're the first people who've been on this block. And that's not really fair either. So that's my long-winded answer to that, Ariel.
I would like to raise a quick question regarding differences, benefits, maybe that. Sorry, what I'm trying to ask is the differences between I guess large scale studies and meta-analyses, because we slowly are gaining access to bigger and bigger data sets that combine the resources from more and more labs. And I know these are different motivations, but in a sense, I feel like science is going in that direction. I know even in our university, labs are starting to conform to a standardized procedure, scanning procedure in the MRI, things like that, that will eventually also help very large scale meta-analyses, but also in the shorter range, I think will change the way we interact with large data. So I guess I'm wondering what you think about the line between there and the future of this kind of research, I guess.
I'm going to give you a really frank and honest answer here. I hope that meta-analysis goes out of business. I hope that maybe in 5 years, we don't have to do this coordinate-based nonsense anymore. I started doing this in 2003, and the culture back then about data sharing was entirely different. There was a lot of data hoarding. There was a lot of secrecy. Folks weren't inclined to share data at all. I actually just wrote a review that's currently, a manuscript review that's currently under review that kind of takes a look back at how much this culture has changed and all of the different steps along the way that got us to where we are right now. I'm at Florida International University, and we're an ABCD site. So I've had that front row seat to participating in a large consortium-based project that is all about acquiring data for the express purpose of openly sharing it to the entire community. And I am delighted with how far our community has come in that way. And I really think that the sky is the limit in terms of, like, as you mentioned, harmonizing protocols so that the data are in essence more poolable. Folks are really much more open-minded about the sharing process. And again, it's been a whole culture shift. And as more raw data are available, then there's going to be less of a need to rely on things like harvesting coordinates from the literature to engage in assessing consensus across studies, because you'll have the data available there. So I do hope I'm put out of business at some point. I'm not sure how long that will take. You know, with image-based meta-analysis, that relies on sharing statistical parametric images in NeuroVault. And so far, that hasn't quite caught on in the way that we had hoped we would by now. There's still quite a heavy burden there in trying to access SPMs from authors to do an image-based meta-analysis. I think the future is bright. I think we have a long way to go, but I think we've come so far that I'm really positive about, you know, what the next 10 years might look like. But yes, put me out of business, please.
One of the slides about the image-based meta-analysis describes a problem that there are different names for the same network. So are there any resources that we could look into so that we can use the nomenclature consistently?
Not yet, but OHBM has a task force that's been meeting for, I think, about a year now. It's the OHBM Network Nomenclature Task, and full disclosure, I'm part of that group. We're actually in the process of putting together, basically, first we want to put out a paper that's a statement of the problem and kind of summarizes all of the different viewpoints. Hopefully, you know, the end goal is that we move towards more community-based recommendations. You know, with that particular paper, the slide that you're mentioning, we do provide recommendations at the end of that paper for those frontoparietal networks. But there's a paper by Lucina Uden, Nathan Sprang, and Thomas Yao that tries to put together a more comprehensive nomenclature system. I will link that paper in the Slack channel when we're done here. Right now, it's currently the best place for you to get recommendations on these network labels, but I really am hoping that we can do better and provide an actual system where we release sets of maps with recommended network labels and give you guys an actual usable tool for the community. That's been something that we've been discussing, but you know, it's a hard problem to solve. So we're working on it. But it's an excellent question. Thank you.
To continue the discussion from before, I guess I'm wondering how do you think about this entropy of analysis variation that is also discussed in the NARPS study, and the tension that will always be, because other than the data, there's of course the analyses, and then we can share the data in different parts or using different standard tools, and just so many options. So I guess there will always be tension between the researchers' need to try different things and the community, or the framework's ability to provide raw data or early data.
So I just watched the finale to Loki, and I'm very much in that whole multiverse mode right now, right? And I gotta say that my response to you is that I really embrace the idea of free will, right? I think our goal as scientists are to make the very best decisions that we can. No one is perfect. No one makes the 100% correct decision at any time. But if we try to make educated decisions, and if we are fully transparent about them and the motivation for them, and that means in our manuscripts, we fully describe exactly what we have done. We try to justify it as we best can. And then we put our work out there for the world to see. And we make our data and our code available so that if someone wants to try to reproduce or replicate what we've done, then they have an opportunity to do so, right? I think that's the best we can all hope for. There is no one correct universe because it's all of these multiverses. And so there is no one correct analysis. And your job is to do the best that you can, but to be transparent and open about it in a way that will promote further science, further work, further knowledge. And I think that's the best that we can all hope for, and that's what we should be striving for.
Maybe we can move to the demo part of the presentation. Taylor, do you want to take it away?
Sure, let's get that going.
Alrighty, so can everyone see the demo, the notebook? Great.
Yep.
So you can run this as well. It's on the Jupyter Hub. It's in the curriculum meta-analysis folder, and it's performing meta-analyses. It's a Jupyter notebook. Alrighty, so this tutorial and demo is, no, it still says OHBM. Well, it's about NIMAIR, which, as you've learned, is a Python library for performing meta-analysis and meta-analysis related analyses, such as functional decoding, automated annotation, parcellation, things of that nature. So there are already a wide range of resources out there for meta-analysis. Databases like BrainMap, NeuroSynth, NeuroVault, or algorithms like Ginger Ale, SDM, and MKDA. Those are all implemented across a range of languages. Some are purely GUI based, some are completely closed source, which makes using the appropriate algorithm for a given question difficult. And people tend to just stick with what they know. So our goal with Nightmare is simply to take all of the primary algorithms for meta-analysis and these other tools, or these other implementations, and put them all in one library with a shared API that you can use to choose whatever algorithm is appropriate for your question. So for those of you who might be familiar with scikit-learn, Nightmare is basically cribbing off of scikit-learn's general API structure, namely that most everything is a class that then is initialized with some parameters. For example, sample size, if you want to set sample size for a full database, or a kernel transformer if you're using a coordinate-based meta-analysis. And then you fit your class, your created object, to a data set to produce a result. Alternatively, we also have transformer classes where you apply a transform method to the data set or to a result and return a transform version of the same thing. So I'm going to go through how we work with data sets, searching those data sets, performing a couple of meta-analyses, and performing some functional decoding. And I guess feel free to jump in if you have questions, or if something doesn't work on your side. So to start, we're going to import everything. We're setting a data directory for where our data are located. And all right, so we have dataset objects, which are loaded from JSON files, which follow a standard we call NIMADs. But for the most part, you don't actually work with these JSON files. You will use something like NeuroSynth format data or BrainMap format data, and we have functions to convert those to Nightmare format. In this case, we have a pane data set from NeuroVault that contains both images and coordinates that we've already converted to the NIMADs JSON format. We can load it into a dataset object with just initializing with the JSON file. And in this case, we're going to set a mask because this data set is in a different space than the standard M&I space. From there, we download the actual images from NeuroVault to our data directory, and we set our path to say that the images are in this location. So this JSON file actually just contains relative paths to where the images will be once you've downloaded them. Once we've run this, we see that the data set has 21 experiments and it's an M&I space. All right. So this contains a number of attributes, including IDs, which is a list of the identifiers for each of the studies in the data set, including a study identifier and an experiment identifier. So the study identifier comes first, then a hyphen, then an experiment identifier. In this case, all of the studies only have one contrast or experiment, so they all just end with -1. But in something like BrainMap, where the studies, the papers are broken up by individual contrasts within the study, then you can have multiple contrasts per paper. There are a number of attributes associated with the data set. Most of them are stored as pandas data frames. They contain relevant information of different types. So for example, coordinates. is a data frame where each row is one of the coordinates for one of the studies. So there are a number of coordinates for each study, which we have in XYZ format. That's just your standard space coordinates, and then converted to I, J, and K matrix indices. We have metadata. This contains any metadata about the study. In this case, all we have is sample sizes. So this would be the sample size of each of the groups in each of the contrasts. So it's a list of integers. And these one sample tests, these are just one integer, one sample size. And then for our images, we have a range of image types, including beta images, standard error, T statistic, Z statistic. And those have both full absolute paths and relative paths. And you'll notice that the Z statistic maps are missing for a number of the studies. That's because with this NeuroVault collection, they only had a subset of images for each of the studies. We have an annotations data frame. This generally contains labels or annotations applied to the data set, to each of the studies in the data set. So in BrainMap, that might be labels defined from the cognitive paradigm ontology applied by individuals to the data set or to the studies in the data set. In Neurosynth, you generally take the text, so the abstract from each study, and you extract a bunch of term weights for each of the studies based on those abstracts. And that's generally how you would do a meta-analysis. In Neurosynth, you just select studies that have, say, the word amygdala in the abstract at some point. In this case, we don't have any annotations, and we also don't have any text, but there's a data frame for text if you want to include, say, the full abstracts or the full text. Now, a couple of other important attributes are the masker, which is it essentially defines the space in which the images in the data set are stored and in which the coordinates will be applied or transformed into image space. We also have a space attribute. This is just a string that describes a space. It's not used for much, but it's a good heuristic to tell if you're in M&I space or something different. Okay. So we have a number of methods that can be used for data sets, including just to save to pickle files. So that way you can load them up again without having to go through the JSON file again. We can also download some of the larger data sets. So we have a fetcher for Neurosynth, and then you can convert these formats to nightmare format with a convert X to dataset function. In addition to so this one we're not going to run because it takes a while to actually run through and it would involve downloading data, but we have it already pre-generated into a pickle file so we can load it up. And we also have a sleuth-based data set. So this is a text file generated by BrainMap's sleuth function or sleuth tool, with which you can search the BrainMap database and export text files to run meta-analyses on. All right. So now that we've loaded up these 3 data sets, this is a sleuth-based one, the Neurosynth data set, and the pain data set, which has images and coordinates, we can try searching them. Neurosynth's the biggest one, so we're going to try using that. That has a number of annotations. In fact, let me just... So in this case, Neurosynth comes with all these TF-IDF values for different words from the abstracts, and they're stored in the annotations attribute. So we can identify studies that have this attribute. So studies with the label pain with a threshold of 0.001, which basically just corresponds to, does this word appear in the abstract at all? And then we can use the slice method to take the IDs that have that match that criterion and reduce the data set to only include those studies. So in this case, we search for pain and we find that there are only 516 out of the 14,000+ studies that have pain at that in the abstract, basically. All right, so now that we can search for studies and slice up those studies to create new data sets, we're going to apply that or we generally would apply that in a meta-analytic coactivation modeling analysis, also known as a MACM. This is where you would identify studies that report at least one coordinate near an ROI of interest, a region of interest, and then run a meta-analysis on just those studies. And that's generally considered like a pseudo functional connectivity analysis applied to meta-analytic data. We have a method called get studies by coordinate, which you put in the coordinate you want and a radius you want to identify studies with at least one coordinate near there. And then you can slice it to create a subset of, or a new data set with a subset of studies. In this case, there are 665 experiments with at least one coordinate within 6 millimeters of 24, -2, -20. All right. So NIMAIR includes implementations of a range of meta-analytic algorithms, including coordinate-based methods like ALE, MKDA, KDA, as well as a bunch of image-based methods. The coordinate-based methods that we have implemented in NIMAIR are all kernel-based. What that means is that they take the coordinates from each of the studies and convolve them with some kind of kernel to generate a pseudo statistical map that can then be combined across studies to perform a meta-analysis to evaluate the significance of or how close all of the coordinates from those individual studies appear, and if there's greater than chance convergence of those coordinates. So in NIMAIR, we separate the kernel from the actual algorithms. That way you can swap out different kernels with different algorithms if you want. There's no guarantee that they'll actually work together well, but you can do it if you want to. We have 3 different kernels, an MKDA kernel, KDA kernel, and an ALE kernel. And each of them have their own parameters that you can define. In this case, we're going to use a radius of 10 millimeters for these two. And we're going to set a sample size of 20 millimeter or of 20 subjects for the ALE kernel, because the distribution of the kernel depends on the sample size. All right, and we can run this. We initialize the kernel, and then we transform our data set using the kernel to produce a list of images. And those images, we're just going to plot the first one. So here we see that.
Do you mind if we jump in with questions here?
No, please go ahead.
There was a question on Slack, and I'm sort of wondering also the word pain in the query that you did. You had sort of TF-IDF __ pain. Can that be replaced by any word?
Yeah. So let's see, where is it?
A bit lower down.
Right here. Yes. So each of these labels in here, you can use any of those. And we just have this neurosynth TF-IDF __ because we want to support multiple sources of annotations. So to make it a little bit easier to understand, in neurosynth, if you go to the neurosynth website, you will see you can do term-based meta-analyses or you can do topic-based meta-analyses. Term-based meta-analyses just take the abstract and they find words or unigrams and bigrams, meaning 2-word terms that appear in those abstracts. The topic meta-analyses correspond to a range of latent Dirichlet allocation topic models that were run on those abstracts. And so you have anywhere from 50 to 400 topics, and each of those would have their own column in this annotations data frame that would say neurosynth, say LDA 50, topic 1. So just make sure that you can search both of those. The But yes, you could use any of the labels that are actually in the data set. And you can get those labels with the get labels method. And you see a list of all those labels. Okay, you know what, that's too many. I'm gonna cut that short. Okay. All right. Any other questions? So we applied our kernels to the pain data set, and we're just looking at the modeled activation or MA map for the first study in that data set with each of the 3 kernels. And you'll see that the kernels look very similar. The modeled activation maps look very similar across the kernels, but they are still different. MKDA kernel, what it does is it just convolves each coordinate with a sphere and then takes the union of those spheres across coordinates within a study. So the map itself is binary and it's either 0 or in this case 1. The KDA kernel, on the other hand, does the same thing with the convolving each of the coordinates with a kernel. And then instead of just taking the union of those coordinate-based maps, it adds them together. So when 2 spheres overlap, you get the sum of them. And in this case, the map has values of 0, 1, 2, or I believe 3, where 3 kernels or when 3 coordinates are close enough together. And then an ALE kernel simply convolves them with a Gaussian distribution that varies in the full width half max based on the sample size. And it takes the maximum value from overlapping kernels. Okay, so these now we have the meta-analytic estimators, such as in this case we're going to look at ALE. And what they do is they take a number of parameters. Most of the time, the defaults are fine. They can also take a kernel transformer in this step, like kernel transformer equals ALE kernel. And if you wanted to feed in a different kind of transformer, you could here, like KDA kernel, MKDA kernel. You can also feed in an initialized one where you have already set the parameters, like this one. In any case, you initialize it and then you fit it to a data set, the new object you fit to a data set to produce a meta result object. Oops. There. All right, so the meta object is a meta-analysis, an ALE object with, and it shows what attributes have been set, what parameters have been fed in. And the meta result object is it's a meta result object. This is really just a light wrapper on a dictionary. So the meta result object contains a maps attribute right here. That attribute is a dictionary with a number of maps that were calculated as part of the meta-analysis. In this case, we have a z-statistic map, a p-value map, and a statistical or a stat map, which is really just ALE values. You can get the map with a get map as a nibabble image, which then you can, as you see, it's a nifty image, and then you can plot that image or save it or what have you for use in your analysis. So if you'll note that this is still kind of, it looks like the coordinates that were convolved, right? Even after thresholding, and that's because no multiple comparisons correction was applied. And in NIMARE, what we do is we separate out multiple comparisons correction from the actual meta-analytic estimation or estimator as an FWE or FDR corrector attribute or class. These classes have a method called inspect, which you can feed in a meta result object, and it'll return the valid methods of correction that are possible with that meta results object and that corrector. In this case, there's a generic meta FWE correction called Bonferroni Correction. Basically, it takes all of the coordinates in the, or all of the voxels in the mask and just adjusts the p values and the z statistics based on them, dividing by the number of voxels in the mask. But there's also a estimator specific method called Monte Carlo, which is what is implemented in BrainMap for Ale. And what that one does is it basically implements runs a Ale specific approach to multiple comparisons correction that involves a range of simulated data sets and building a null distribution of values from those simulated data sets. So now that we know Monte Carlo is a valid method for this meta result object, what we do is we initialize our corrector with the method we want, in this case, number of iterations and number of cores we're going to use, which I will set to 2 for speed. And then we can transform our meta result object to produce a new updated meta result object. So this is going to run 100 iterations of the Monte Carlo simulations to produce a null distribution of cluster sizes as well as Ale values, with which voxel and cluster level FWE correction can be done. And our new meta result object still has a maps attribute, but as you can see, there's a number of new maps that have been added. So we have our stat P and Z maps. Those are still exactly the same as they were before, but now we have log P and Z maps for voxel level correction, as well as log P and Z maps for cluster level correction. And we can look at our cluster level corrected Z statistic map, which for each cluster, there is a p-value across the whole cluster. You could threshold this based on your p-value, your alpha value that you want to set it to, and report that in your paper. You can also run get clusters table from Laird, which will output the peak coordinates and cluster sizes, although it behaves a little bit oddly when each of the clusters is just one value like this. Okay. So all of the different coordinate-based meta-analyses operate basically the same way. You could swap out most of the code here with an MKDA meta-analysis, an AL meta-analysis, scale meta-analysis, or a KDA meta-analysis. So we'll move on to image-based meta-analysis. This is the full...
For that, there's actually a question from the audience. Harvey, go ahead.
Hi, yeah. My question is, I changed the cut coordinate to 0, 0, 20, -24, like the maps that were plotted earlier, but it's not the same heat map. I know that we're using the same pane data set, so I'm wondering what the difference is between what we're seeing in the slices above and what we're doing here.
Sorry, so you're talking about up here in the kernels?
Yeah, right there at those Ale kernels. Why is that different than the one below, even if you change to the same slice?
The one below, which, down here?
Yeah.
This? Okay, so this is a modeled activation map. Basically, it's just the coordinates from one of the studies in the data set convolved with the kernels. So these are essentially the inputs to our meta-analysis algorithm. In this case, it's going to be Ale. The Ale algorithm essentially just combines these MA maps to produce this one. So this is combined across all 21 studies in that data set.
Gotcha. Great. Thank you.
And then from there, you would apply multiple comparisons correction because an uncorrected statistic map is just not reportable. Okay, so image-based meta-analysis, we have all of our images from the 21 studies in the pain data set here. And you'll note beta maps, standard error maps, T maps for all the studies, but Z maps only for the last 11 or no, 10 or so. So in this case, if you wanted to do a meta-analysis that relies on Z statistic maps, you could only do a meta-analysis of 10 or no, 11 studies. However, NIMER has an image transformer class that actually will take existing images, if the necessary images are available, and convert them into target images. In this case, we have T statistics and degrees of freedom, so we can get Z statistics from our T statistic maps. And we also have standard error maps, which will allow us to calculate varcope maps, which is it's just another measure of the variance. And it's necessary for a number of image-based meta-analyses. So now that those have been transformed, you can see our images attribute now has images for the Z statistic maps for those first several studies. And if we scroll all the way to the right, varcope maps have been added as well. So there are a wide range of image-based meta-analyses that you can do, and they vary based on what data you have. So to start, we'll just look at a hedges meta-analysis. This one uses beta and varcope images. And it just, you initialize it just like you did with a coordinate-based meta-analysis, and you fit it to the data set. And when it's fitted, it will grab the relevant images and apply the meta-analysis to those images. And this produces, we can actually take a look at the meta result object. All right, so this produces 4 maps. Tau squared, which is the estimate of between study variability, Z statistic p-value, and estimate, which is the essentially the mean across studies of the beta values going in, scaled by the variance. All right, and we've plotted our Z statistic map from this. And as you can see, it's a lot more rich. There's values at every voxel compared to the coordinate-based meta-analysis above, which is much more sparse because coordinates are, a reduced version of the information from the study, from the maps. All right. We also have a permuted OLS method. This uses Z statistic maps from the data set. And it fits very quickly. And the map looks very similar. Obviously, the scaling is quite different and the actual values are very different. But the distribution of values looks very similar. And then we have a special multiple comparisons correction method for the permuted OLS, which is also called Monte Carlo. We will define our corrector just like we did before for the ale, and we will use it to transform the meta result object to produce a new meta result object. This takes a moment because we're running 100 iterations, but not nearly as long as the ale did. And this produces, so this has T, Z, log P, and Z maps, or T and Z from the original meta-analysis, log P for voxel level corrected, and Z for voxel level corrected maps produced by the multiple comparisons corrector. If we render the actual corrected map, you'll see that it's a lot more sparse. A lot of values got pushed, or a lot of Z values got pushed down to 0 or near 0 because of the correction. We can also run get clusters table on this to produce a table that you might put in a manuscript. And yeah, it looks like there are a large number of clusters, 13 or so. Okay. So here, all right. So back in 2014, Camille Momet and Tom Nichols had a preprint where they had an SPM image-based meta-analysis extension, and they showed results from that. Here are a few of the results from a few of the different algorithms that they had implemented there. And as you can see with our own algorithms, they look very similar, or at least the image-based meta-analysis does. The coordinate-based one, you can see that some of the peaks or some of the significant clusters are very similar, but obviously much reduced because the power is just much lower for a coordinate-based meta-analysis, especially when you're only running it on 21 studies. Okay, so that's image-based meta-analysis in NIMAIR in a nutshell. Any questions about that? Okay. One of the other big things you can do with NIMAIR is functional decoding. So this is where you try to infer, you basically use imaging data to infer function, specifically using annotations from a meta-analytic data set. So with NeuroSynth, we would probably use those TF-IDF values that we saw above. And we could apply a decoding method to a region of interest, a continuous map if we wanted, or we could even use encoding methods which try to take text and infer or convert it to a simulated statistical map, which is what NeuroQuery does. So we're going to skip over continuous decoding methods because they take a very long time and involve creating a large number of images. But basically, if you wanted to do a continuous decoding, what it would involve is running a meta-analysis on each of the terms in the data set. So for NeuroSynth, that would be those 3,000 or so labels above. Run a meta-analysis on each of them, save one of the maps from that meta-analysis, and then take your continuous map that you want to decode and correlate it with each of those maps to produce 3,000 or so correlation coefficients. In practice, that takes such a long time that it's not worth doing, at least not in the tutorial here. What we are going to talk about is discrete decoding methods, which will generally take either a region of interest or some other selection criterion to identify a subset of the data set that you want to analyze. So in this case, well, first, I guess I should say there are two main methods. That's the brain map decoder and the NeuroSynth decoder. Both of them work fairly similarly. You can see a bit more about the actual algorithms here, but they essentially work by taking a data set with both annotations and coordinates, splitting up that data set based on some criterion that you want to apply, like does a study report coordinates within an ROI? And then comparing the annotations from that subset to the rest of the data set. You can get both forward inference values, which essentially just evaluate the subset, the selected subset of the data set that you want to look at. And you can get reverse inference ones, which compare that subset to the remaining part of the data set. To move things faster here, we're just going to run it on the first 500 studies in the NeuroSynth data set. And we're going to set our criterion as, did the study report the word amygdala in its abstract? That's not really a meaningful search criterion if you actually want to do functional decoding, but in this case, it's just easy to run. All right, so of the first 500 studies, there's only 38 that have this amygdala label. Each of these decoders works a lot like the other classes in NIMAIR. What you do is you initialize with parameters. In this case, we're going to say we don't want to do multiple comparisons correction. And then you fit that to a data set. Now, this one also has a transform method, which then you use to transform the sample that you want to look at. This is the list of studies that report amygdala in the abstract to produce a data frame that has one row for each of the labels in the study or in the data set, and 6 or so columns, one for each of the measures that you're getting out from this decoding method. And NeuroSynth Decoder does the same thing. And in addition to these two, which are still running, we have an Roi Association Decoder, which I just added this past week. It took a method from NeuroSynth, wherein what it does is it just takes a region of interest mask. It calculates modeled activation maps for all of the studies and averages those modeled activation values within the region of interest mask to get a one-dimensional array of MA values. And then it correlates that with the TF-IDF or whatever annotation values for all of the different labels in the data set. All right, so for the brain map decoder, we get out one row for each of the annotations and 6 columns. These are forward inference p-value, forward inference z-statistic, the likelihood for the forward inference analysis, reverse inference, p-value, z-statistic, and posterior probability. Same thing for the NeuroSynth, except we have a posterior probability for the forward inference instead of a likelihood value. And they essentially try to do the same thing, although they have different implementations, different algorithms. And so what you would probably want to do is sort by, say, reverse inference posterior probability, which we did here, and see which labels are most associated with your search criterion. In this case, amygdala obviously shows up as the first because that was our search criterion. There are a bunch of generic terms as well as emotional, response, neutral, etc. It would be a lot more meaningful if we ran it on the full NeuroSynth database. With the NeuroSynth method, we get amygdala, amygdala response, amygdala hippocampus, etc. And then for the ROI association decoder, it's just a series of correlations. So we get correlation coefficients, but you can see that they are also pretty meaningful. Amygdala fear reinforcement appraisal. Here's an exercise which we really don't have time to do. But if you want to, you could, since you'll have access to the notebook. The idea is just to run a MACM and then decode an ROI using the data sets that are already here and a region of interest mask right here. All right. Does anyone have any questions?
I'd like to ask whether there's any implementation of something similar to a scikit-learn pipeline for this kind of workflow.
No, we have, so this is not scikit-learn-esque at all. We have a couple of command line interfaces that string together these a few common steps into one workflow. They also generate citable boilerplate text describing what happened. But no, there's no built-in tool to string together different steps of a meta-analysis workflow. It's a good idea, though.
I also just wanted to say it's a really impressive demo and amazing work. It's really inspiring stuff.
Thanks.
You know, having a combined or having a single place where you can access all of these different algorithms, you know, Taylor made a point of saying that, but like coming from the historical perspective of watching this group develop this algorithm and this other group develop this other algorithm, and they've always been completely separate, and you've always had to, if you want to switch from one to the other, you know, basically start back over and do everything from scratch. And so, you know, when Taylor gets to the point in the tutorial where he was like, you can do this one, or you can just change it and do that one, like my brain explodes when he does that, because It's just so different from how things started, and it shows the beauty of the evolution of developing these platforms and these tools where everything is all sort of integrated and open and available. And the point that was made about the workflow, if that's something that's of interest to you, then come on in and help us develop it. And I think that sort of framework and collaborative mindset is what makes all of this so exciting.
Thank you. I just might. I'm working on I hope things that could associate with it, and yeah, that would be great if they would.
Are there any other questions? I notice we're at the end of our hour here, so I think that was timed really nicely, but anything else that anybody wants to throw at us? Well, we appreciate the opportunity to be here and to share the stuff that we've been working on, and we hope you have a good rest of the week. Taylor and I are on the Slack. I'm actually checking out for a holiday. I will try to check back in if there are any messages, but I will get to them eventually if I do miss them. Taylor will be here and able to catch things more on the fly. But good luck with the rest of the week. And if you need anything, just reach out.
Thank you so much. Thanks for the presentation.
Thank you.
And the tutorial and everything. Take care, everyone.
Thank you very much.
Thank you.